Hi everyone, my name is Melissa Rivera Lopez and I'm a PhD student at Dr. Demetrio Sierra's lab. Of the many projects we have in our lab, today I'm going to be talking to you about the effects of atrazine on fear behavior in rodents. So let's learn a little bit about atrazine. Atrazine is an agricultural herbicide and it works by inhibiting photosynthesis. Therefore, it's considered safe for mammals. However, given its extensive use worldwide, it has been detected in food and drinking water, and therefore it's considered an environmental contaminant. Now, rodent studies have shown that ingestion of high doses of atrazine can increase anxiety-like behaviors. But unlike with other agricultural chemicals, studies of atrazine looking at fear behaviors and their related brain regions remain largely unexplored. Therefore, the main problem I wish to point out here is that rodent studies have already shown that exposure to atrazine can influence anxiety-related behaviors, but other behaviors that are also emotionally relevant, such as fear, have not been looked at, and that's what we aim to do here. Now, the significance is that humans are also exposed to atrazine through their drinking water and also in certain foods. In fact, clinical studies of herbicide exposure has shown that it can disrupt brain function in humans. Therefore, this makes atrazine a potential public health concern where it may lead to emotional disorders in humans. So our hypotheses for these experiments are one, that atrazine exposure will increase anxiety-like behavior in adult male rats, and two, that it will also increase fear behavior in adult male rats. So for our methods, we use the oral reference dose as the level of atrazine exposure, which is 0.035 milligrams per kilograms, and this reference dose is established by the Environmental Protection Agency and is considered safe for human consumption. And our animals were exposed to atrazine through their drinking water. Now, here is a timeline that shows the period in which they were exposed to atrazine and the different tests that were carried out. So after 3 months of atrazine exposure, they underwent the open field test, and after 4 months, they underwent fear conditioning. After these behavioral experiments, the animal's brain tissue is collected and will be examined for changes in neuronal activity using CFOS immunohistochemistry. Now, for our first paradigm, we used the open field test following 3 months, and with this we wanted to look at anxiety-like behavior. So here, we're basically showing an open space where the animal is placed, and a healthy animal would, explore the entire area. And in general, rodents do prefer corners and the periphery, but from time to time they will explore the center area. And this was done with each animal for a total of 5 minutes. So as you can see here, this is just a tracking plot of the animal exploring the open field tests. Now, what we really look at in this paradigm is how much time the animal spends in the center of an open field versus the periphery. And that is the parameter that we use to examine anxiety-like behavior, where if the animal spends less time in the center and more time in the periphery, that animal is considered to show anxiety-like behavior. Now, we'll see what that looks like when comparing both groups. So what you're seeing here is in a sham animal, thus showing a relatively healthy exploratory behavior. So our findings show that the atrazine exposed animals actually spent more time in the center, compared to the sham animals, and this was significantly different. So this means that thus far, atrazine appears to increase time in the center, which is consistent with decreased anxiety instead of increased anxiety, interestingly enough. After this paradigm, we went and ran another behavioral test following 4 months, which was Pavlovian fear conditioning, and this was used to measure fear behavior. So in Pavlovian fear conditioning, the animal is placed in a sound attenuated chamber, and as you can see, he's just going about his business, but there's going to come a time where a tone is going to appear, and at the end of the tone, that animal is going to receive a shock. Okay, that means you see that red symbol, that means the tone is on, and at the end of that tone, he's going to receive a shock. He wasn't expecting that, okay? Now, if we expose the animal to that same tone 24 hours later without the shock, we're going to see what type of behavior it exhibits. As you can see, the video isn't paused or anything. The animal is just showing freezing behavior. That means that the animal learned that the tone predicts shock. Now, if we continue to show the animal that same tone without the shock several and several times, the animal is going to learn that the tone no longer predicts danger. So it learns something new. It learns to extinguish that behavior, as you saw there, okay? So the animal, during extinction, he hears the tone and no longer freezes, and that's considered extinction. Now, let's see what that looks like in a graph. Let's see how our atrazine animals did thus far. So as you can see here, during conditioning, where the animals received the tone and shock, both groups made that tone shock association without any trouble, so all animals learned that the tone predicts a shock. Now, in subsequent days, where they were exposed to the tone without the shock several times, they learned something new. They learned that the tone no longer signals danger, and therefore they extinguish that freezing behavior, which is our index or measure of fear. So as you can see, in the next 3 days of extinction, there's no significant difference between any of the groups. Therefore, atrazine does not appear to influence fear or extinction memories with our animals. Now, in summary, what we've seen so far is that atrazine exposure actually decreases anxiety-like behavior in adult male rats, and thus far it has no effect on fear behavior in adult male rats. So we conclude that atrazine exposure at a dose considered safe for human ingestion, such as the oral reference dose used here, does not affect fear behavior, but appears to have actually an anxiolytic effect. Now, even though we didn't see any fear-related impairment or even though there was no significant difference regarding fear behaviors, we may still see some differences in neuronal activity. If we look at the brain regions necessary for fear behaviors, which would include the amygdala and the hippocampus. So as part of our future directions, we are going to increase the number of animals in the study and complete tissue analysis for neuronal activity to compare it to behavior and to brain function. So in this case, you can see that one of the brain regions important for fear is the hippocampus, shown in this section. And what it points to is a magnification at 4 and 10x of that tissue stained with CFOS to look at changes in neuronal activity. And this is data collected at our lab. So this is currently underway, and hopefully we will have that data soon enough. Now, given that this is the end of my presentation, I wish to appreciate all the funding agencies that have made these experiments possible, as well as our team at Sierra Mercado Lab, which are all the team members are shown here in this picture. So, with this, I conclude my presentation and I'm ready to answer any questions. Thank you very much.